Tes 1 2 3. Mbak Ana izin, suara saya sudah terdengar dengan baik, Mbak Ana?
Sudah, Mbak.
Oh, baik, terima kasih, Mbak. Ya, selamat pagi. Terima kasih kawan BRIN yang sudah bergabung, baik melalui media Zoom maupun kanal YouTube BRIN Indonesia. Silakan untuk mengisi presensi Bapak Ibu ya, nanti yang akan di-share oleh panitia di pertengahan acara. Sebentar lagi acara akan kami mulai. Kembali saya menyapa, terima kasih kawan BRIN yang sudah bergabung, baik melalui media Zoom maupun kanal YouTube BRIN Indonesia. Baik Bapak Ibu, mungkin acara sudah bisa kami mulai. Kita mulai, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera bagi kita semua. Yang terhormat Kepala Organisasi Riset Kesehatan Profesor Dr. Hewani Luputu Indi Darmayanti MSI. Yang kami hormati Kepala Pusat Riset Biomedis Profesor Dr. Sunarno MSI Med, dan yang kami hormati para narasumber pada pagi hari ini, yaitu Profesor Leo Kokwen, Researcher Professor dari Department of Medical Research Hualin Suzy Hospital. Anggreni Dini Pasiana PhD, peneliti ahli muda Pusat Riset Biomedis OR Kesehatan BRIN, serta para tamu undangan, peserta webinar, dan kawan BRIN yang berbahagia. Alhamdulillah, puji syukur kami panjatkan karena berkat rahmat Allah Subhanahu wa Ta'ala, Tuhan yang Maha Esa, kita dapat berkumpul untuk menghadiri acara sharing session pada acara Neuroscience Talk Session 1 Current Research Clinic Innovation and Future Perspective, Selasa 1 September 2026. Baik Bapak Ibu hadirin yang kami hormati, mengawali acara hari ini menyanyikan lagu kebangsaan Indonesia Raya. Hadirin dimohon untuk menyimak dengan hikmat. Baik, terima kasih Bapak Ibu. Cara selanjutnya yaitu sambutan Kepala Organisasi Riset Kesehatan, Profesor Dr. Hewani Putu Indi Darmayanti MSI. Kami silakan.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sehat bagi kita semuanya. Yang terhormat Profesor Dr. Sunarno selaku Kepala Pusat Riset Biomedis BRIN, para narasumber, Dr. Hopkin Liu, researcher from the Department of Medical Research, Hualien Suzy Hospital, Ibu Agriani Dini Pasiana, PhD, peneliti ahli muda dari Pusat Riset Biomedis BRIN, para akademisi, klinisi, peneliti, dosen, mahasiswa, serta seluruh peserta Neuroscience Talk Section 1 yang berbahagia. Puji syukur kita panjatkan kehadirat Allah Subhanahu wa Ta'ala karena atas rahmatnya kita dapat hadir pada kegiatan webinar dengan tema Current Research, Clinical Innovation, and Future Perspective. Hadirin yang saya hormati, saat ini penyakit neurologis menjadi salah satu tantangan kesehatan global yang semakin penting. Beban penyakit neurologis terus meningkat seiring pertumbuhan dan penuaan populasi. Global Burden of Diseases tahun 2021 menunjukkan bahwa gangguan neurologis secara keseluruhan memberikan kontribusi besar terhadap kematian dan disabilitas di dunia. Dalam konteks tersebut, perkembangan neuroscience menjadi semakin penting. Kita tidak hanya dituntut untuk memahami manifestasi klinis penyakit, tetapi juga menggali mekanisme biologis dan molekuler yang mendasarinya. Pemahaman tersebut menjadi dasar untuk menemukan pendekatan baru dalam diagnosis, pencegahan, terapi, dan rehabilitasi. Kemajuan neuroscience saat ini juga menunjukkan bahwa masa depan kedokteran akan semakin mengarah pada precision medicine, integrasi basic science dengan clinical research, serta pemanfaatan teknologi molekuler dan digital untuk menghasilkan intervensi yang lebih efektif dan personal. Melalui kegiatan hari ini, kita berharap dapat mempertemukan berbagai perspektif dari penelitian dasar hingga inovasi klinis, serta membuka peluang kolaborasi antara akademisi, klinisi, peneliti, dan juga generasi muda. Tema Current Research, Clinical Innovation, and Future Perspective juga mengingatkan kita bahwa penelitian bukan hanya tentang menemukan sesuatu yang baru, tetapi bagaimana penemuan tersebut dapat diterjemahkan menjadi solusi yang nyata bagi pasien dan masyarakat. Forum hari ini menjadi sangat istimewa karena menghadirkan dua perspektif penelitian neuroscience yang sangat menarik dan relevan dengan perkembangan ilmu pengetahuan saat ini. Kehadiran kedua narasumber dengan latar belakang penelitian yang kuat diharapkan dapat memberikan perspektif yang lebih luas mengenai bagaimana penelitian molekuler dapat menjadi dasar bagi pengembangan neuroscience di masa depan. Saya berharap kegiatan ini dapat menjadi ruang untuk bertukar pengetahuan, membangun jejaring ilmiah, menghasilkan gagasan penelitian baru, dan memperkuat kolaborasi neuroscience di tingkat nasional maupun internasional. Akhir kata, saya menyampaikan apresiasi dan terima kasih kepada seluruh narasumber, panitia, dan peserta yang telah berkontribusi dalam kegiatan ini. Dengan mengucapkan Bismillahirrahmanirrahim, kegiatan Neuroscience Talk Section 1, Current Research, Clinical Innovation, and Future Perspective secara resmi saya nyatakan dibuka. Selamat mengikuti kegiatan. Semoga forum ini memberikan manfaat dan menjadi langkah menuju kemajuan neuroscience serta pelayanan kesehatan neurologis yang lebih baik. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih. Selanjutnya mohon kesediaan Bapak atau Ibu untuk foto bersama dengan menyalakan kamera dan memberikan senyuman terbaiknya. Kita akan memulai sesi foto sesuai dengan aba-aba saya. Panitia akan melakukan tangkap layar pada layar panelis. Baik, pada mulai hitungan ketiga, kita akan mulai sesi foto pertama. 1, 2, dan 3. Baik, selanjutnya akan ditangkap layar oleh panitia. Terima kasih Bapak dan Ibu. Selanjutnya memasuki acara inti, yaitu pemaparan dan diskusi yang akan dipandu secara langsung oleh moderator kita hari ini, yaitu Jiro Hesagawang Sitomorang PhD, Satuan Kerja Peneliti Pusat Riset Biomedis OR Kesehatan BRIN. Baik, langsung saja kami persilakan kepada Jiro Hesagawang Sitomora untuk memandu sesi paparan dan diskusi. Waktu dan layar dipersilakan.
Yeah. Thank you to Bu Rin. Can you hear my voice?
Yes.
Okay. Thank you, everyone. Thank you for our MC, Bu Rin, for handing over the floor. And special thanks to our head of Organization for Health, Professor Indi Darmayanti, for those inspiring welcome remarks. And for everyone, good morning. Welcome today to the Neuroscience Talk Section 1. And our theme today is the current research, clinical innovation, and future perspectives. My name is Jiro, and I'm your moderator today. And it is an absolute honor to be your moderator. Here, I'm a researcher at the Center for Biomedical Research. Today, we have two outstanding speakers joining us today to share their research. Now, before we begin our talk, a quick reminder to everyone, to our audience, please type your question later in the chat box, in the Q&A box, or chat at any time during the presentation. And we will address your questions during the discussion session at the end of the speakers too. And let's get started with our first speaker, Professor Liao Hokkien. Professor Liao Hokkien earned his PhD from the National Defense Medical Center, and now he serves as a researcher at Hualien Tzu Hospital, a position he held since 2015. Professor Liao has extensive work in intracerebral hemorrhage, and with recent publications features in top-tier journals such as Cell Death and Disease and Experimental Neurology. And today, Professor Liao will be presenting his research. The title is From Hematoma Burden to Brain Repair, Enhancing Microglial Clearance and Resolving Neuroinflammation After Intracerebral Hemorrhage. So without further ado, let's welcome Professor Liao. Professor Liao, you have around 30 minutes for your presentation. The floor is yours. Thank you.
Okay, thank you, Jiro. Can you hear me?
Yes.
Okay. Yes.
Very good. All good.
Okay. Okay. Do you see the PowerPoint?
Yes, Professor Liao, everything.
Okay. Good morning, everyone. Selamat pagi. Thank you for the kind invitation and for giving me the opportunity to share our work today. My name is Hokkien Liu from the Department of Medical Research at Hualien Tzu Hospital in Taiwan. Today, I would like to talk about our recent work on intracerebral hemorrhage, or in short, ICH. The title of my talk is From Hematoma Burden to Brain Repair, Enhancing Microglial Clearance and Resolving Neuroinflammation After Intracerebral Hemorrhage. Rather than focusing only on neuronal death after ICH, today I would like to discuss another perspective. How can the injured brain actively remove the hematoma, resolve inflammation, and eventually enter a state that is more favorable for repair? In particular, I will focus on the role of microglia and macrophage in this process. So let me begin with a brief overview of why intracerebral hemorrhage is method. As we all know that the stroke can generally be divided into ischemic stroke and hemorrhagic stroke. Ischemic stroke account for the majority of cases. However, intracerebral hemorrhage represents only around 10 to 50% of all stroke, but it causes a disproportionately high degree of mortality and disability. Early mortality remains very high, approximately 30 to 40%, and hemorrhagic stroke contribute to millions of deaths worldwide. Another important point is that the geographic difference compared with Western populations, hemorrhagic stroke has a relatively higher incidence in many Asian populations like Taiwan, Indonesia, and Malaysia also. Compared with Western population, I think throughout the ICH is less common than ischemic stroke. The clinical burden is extremely high. Anatomically, hemorrhage commonly occur in deep brain regions, such as the basal ganglia and thalamus, cerebellum, and brainstem. And once bleeding occurs, the hematoma itself immediately becomes both a mechanical and biochemical sources of brain injury. These bring us to the first clinical questions. Once we identify a hematoma, what can we actually do for the patients? Clinically, the management of ICH depends strongly on hematoma size, location, neurological status, and overall disease severity. For patients with relatively mild to moderate hemorrhage, treatment is often supportive, including blood pressure control, management of intracranial pressure, and close neurological monitoring. In this patient, we largely depend on the brain endogenous mechanism to gradually absorb the hematoma. However, in patients with severe ICH and significantly mass effect, more aggressive intervention may be required. This approach includes conventional craniectomy or craniectomy, craniotomy, and minimal invasive hematoma aspiration. And in selective cases, external ventricular drainage also can be applied. But there's one important limitation. Even after surgical evacuation, we rarely remove every component of the hematoma. Residual red blood cells, hemoglobin, heme, iron, and damaged tissue remain inside or around the hemorrhagic brain. Therefore, whether the patient receives conservative treatment or surgical evacuation, hematoma-related secondary injury continues to be a major problem. So in this slide, summarize the pathological of ICH immediately after vessel rupture. The expanding hematoma causes primary injury through direct tissue disruption and mass effect. But what happened afterward is equally important. The red blood cell and other blood components began to break down. This released hemoglobin, heme, and many other damage associated molecules into the surrounding brain tissue. This component trigger a complex secondary injury response, including inflammation, oxidative stress, neuronal cell death, blood-brain barrier disruptions, and mitochondrial dysfunction. As shown in the image on the left, even within only a few hours after ICH, red blood cells are already surrounded by activated cells in the perihematoma area. So in many ways, the hematoma gradually changes from a mechanical problem into an inflammatory and metabolic problem. And one of the major cellular populations responsible for responding to this blood-derived material is the microglia. So what is microglia? Microglia are the resident immune cells of the central nervous system. Under physiological conditions that continuously survey the brain and microenvironment, they remove cellular debris, support synaptic remodeling, maintain tissue homeostasis, and communicate with neuron, astrocyte, oligodendrocyte, and vascular cells. Traditionally, microglia were also often classified very simple into M1 and M2 phenotype. But today, we know that this is an oversimplification. Microglia activation actually exists along a dynamic and highly context-dependent spectrum. Their phenotype depends on the type of injury, disease stage, local metabolic environment, and the signal they receive from neighboring cells. So in this talk, when I refer to microglia activation or reprogramming, I am thinking more broadly than the classical M1 versus M2 concept. Microglia can respond to many different types of signals. These include pathogens, cytokines and neurotransmitters, cellular debris, ATP, arrest, and damage-associated molecular pattern. In the context of ICH, many of these signals are present simultaneously. Therefore, microglia can become either beneficial or detrimental depending on their functional state. On the beneficial side, they are clear dead cells, red blood cells, and toxic metabolites, and contribute to tissue remodeling. On the detrimental side, excessive or prolonged activation can produce inflammatory cytokines, arrest, and other mediators that further damage neuron and the blood-brain barrier. This dual role is extremely important. We do not simply want to suppress microglia. Ideally, we want to preserve or enhance their reparative functions while limiting inflammatory toxicity. Okay, here you can see an example of microglia interaction with red blood cells. Following ICH, microglia and infiltrating macrophage becomes activated, proliferate, migrate toward the hematoma, and begin to engulf blood-derived material. You can think about this process as three sequential steps: find me, eat me, and digest me. First, microglia must recognize and migrate toward the damaged material. Second, they need to engulf the RBC. And finally, they have to digest and metabolize that cargo safely. The time-lapse image are illustrated here, this dynamic process. The small particles are red blood cells, while the moving cells are microglia. Importantly, this process is not restricted to red blood cells. Microglia must also deal with many other damaged cellular components after hemorrhage. So, can we enhance microglia clearance without amplifying inflammation? After ICH, red blood cell lysis activate microglia. Initially, the inflammatory response is necessary because the brain has to detect and contain the injury. However, a persistent pro-inflammatory activation can become harmful. At the same time, efficient hematoma clearance requires highly active microglia phagocytosis and metabolism. So this is a biological tension here. We need active microglia, but we do not want uncontrolled inflammatory microglia. Our working hypothesis is that an effective therapy should reprogram microglia toward a clearance-oriented and inflammation-resolving state, rather than simply switching inflammation off. To address this question, experimentally, our laboratory uses several animal models of ICH. The first is the collagenase-induced ICH model. Collagenase disrupt the vascular extracellular matrix and produce progressive bleeding, which is useful for studying continuous hemorrhage and secondary injury. The second is autologous blood infusion ICH model, in which blood is directly injected into the brain. This gives us better control over the amount of blood, and is useful for studying the biological effect of the hematoma itself. We also established a balloon inflation model, which allows us to separate the mechanical mass effect of a hematoma from the biochemical effects of blood. For most of the work I will show today, we use the collagenase model. Collagenase type 7S is stereotactically injected into the striatum. In our commonly used model, we inject 0.2 unit in 1 microliter, targeting the striatum at the coordinate shown here. Mechanistically, collagenase degrades the component of the vascular basement membrane and extracellular matrix. This damage small vessel and result in blood extravasation into the surrounding brain tissue. One advantage of this model is that hematoma formation develops progressively, making it useful for investigating both early bleeding and sequential inflammatory response. After inducing ICH, we do not evaluate treatment only one endpoint. We use a multi-level assessment strategy. First, 7 Tesla MRI allow us to quantify hematoma volume, brain edema, and structural injury. We also evaluate blood-brain barrier permeability. And the second, we assess neurological functions using behavior tests such as the modified neurological severity score and Rota Road and the locomotor activity. This is important because reducing lesion size is meaningful only if it translates into functional improvement. Therefore, our experiment designs integrate anatomical, vasculature, and behavioral outcomes. In parallel with these functional assays, we investigate the underlying molecular mechanisms we use in blood and the immunohistochemistry to examine inflammation, stress response, and phagocytosis-related protein. More importantly, we also can isolate specific cell population from the hemorrhagic brain and perform RNA sequence. This gives us the opportunity to move beyond whole brain measurement and ask, what is the actually happen inside the microglia themselves? This cell-specific approach later become very important for understanding the mechanism I will show you. Based on this platform, our therapeutic concept is relatively straightforward. Regardless of whether hematoma resolution occur naturally or following surgical intervention, the brain must eventually deal with residual blood component. They are interested in pharmacological strategy that we reduce hematoma burden and neuroinflammation by enhancing endogenous microglia clearance. One molecule that became particularly interesting currently to us is the CDNF. CDNF, full name is cerebral dopamine neurotrophic factor. Initially, CDNF was not developed as a hematoma clearance drug. Our original interest becomes from neuroprotective and stress modulating property. But as I will show you, the biological becomes much more interesting than we initially expected. What is CDNF? CDNF belongs to a small family of unconventional neurotrophic factor together with MEMF. This protein contains several structure, interesting regions, including a subposins domain, like an C-terminal region associated with intracellular stress regulation. Actually, CDNF was originally characterized through work led by Professor Mark Salma Finland, and particularly in model of dopaminergic neuronal injury. Unlike classical neurotrophic factor, CDNF has a strong relations with endoplasmic reticulum homeostasis and cellular response in different disease models. In different disease models, CDNF has been reported to counteract with ER stress and support cell survival. This makes us interested in whether CDNF could protect the hemorrhagic brain, where oxidative stress, ER stress, inflammation, and metabolite dysfunction occur simultaneously. Before administering recombinant CDNF, we first ask what happened to endogenous CDNF after hemorrhage. As shown here, CDNF expression in the hemorrhagic striatum decreased during the early stage of the ICH. This is interesting because this early period coincides with intense oxidative stress, inflammatory activations, and blood cells breakdown. Therefore, the endogenous CDNF may be insufficient precisely when the injured tissue is under greater stress. This provides a rationale for testing whether supplementation with exogenous recombinant human CDNF could be beneficial. To further understand the role of CDNF after ICH, we examine ICH injury in CDNF knockout animal. As shown here, compared with wild type, CDNF knockout mice develop a substantially larger hemorrhagic lesion at day 3 after ICH. We then examine whether exogenous administration recombinant human CDNF goes within the brain. We label the CDNF with a fluorescent marker and administrate it intracerebral ventricularly. 6 hours later, labeled CDNF was clearly detectable in the brain around the injury sites. When we examine different cell types, we found colocalization, particularly with neuron microglia, including activate microglia in injured region. This was important because it suggests that microglia could potentially be a direct cellular target of CDNF rather than simply responding secondary to reduced neural injury. We next evaluate the effect of CDNF in both collagenase and blood infusion model. Recombinant CDNF was given intracerebral ventricularly at the doses shown here, 1, 5, and 10. Across the models, CDNF reduced the hemorrhagic lesions and improved neurological recovery. Importantly, the response was not limited to one specific ICH model. This gives us greater confidence that the therapeutic effects were related to biological response to blood-induced brain injury rather than an artifact of collagenous alone. The anatomic improvement was accompanied by functional recovery in NMSS. The higher the score, the more severe the injury. Animals treated with CDNF show a faster reversal of neurological deficit. In the cylinder test, which measured as metric in the 4 limbs used, CDNF-treated animals also show improved performance. In addition, the Turner analysis demonstrates fewer apoptotic cells. So, at this stage of the project, the interpretation seems straightforward. CDNF is neuroprotection, reduce cell death, and improve neurological function. But the next set of experiments suggests that something else was happening. Here we examine several structural components of secondary injury. CDNF treatment reduced lesion volume. MRI also demonstrates a reduction in brain edema. And in addition, vascular permeability and blood-brain barrier disruption with attenuate, as we can see the events blue assay. Taken together, these data support a broad protective effect of CDNF on the perihematoma brain. At this point, one obvious explanation would be that CDNF simply suppresses inflammation. And indeed, CDNF has a strong anti-neuroinflammatory effect. As you can see from these cytokine measurements, including interferon gamma, TNF-alpha, IL-1 beta, and IL-6. CDNF treatment reduced the pro-inflammatory cytokines and restored the anti-inflammatory cytokine IL-10. So initially, we thought this might explain most of the therapeutic benefit. Less inflammation leads to less edema, less secondary brain injury, and better neurological recovery. That explanation is reasonable. This one was the most interesting observation in this project. We measure the residual hemoglobin using the Drapkin assay as an index of hemorrhagic burden. At day 3 after ICH, the residual hemorrhagic volume was significantly lower in the CDNF-treated animals. In other words, the blood itself was disappearing faster. This was unexpected. CDNF was administered after hemorrhage, so it was unlikely simply to prevent the initial hematoma from forming. Instead, the results suggest that CDNF was accelerating hematoma resolution, and this raised a new mechanistic question. How could a neurotrophic factor increase the clearance of blood from the brain? We already know that CDNF broadly shifts the inflammatory environment, but anti-inflammatory side activity alone does not explain why hemoglobin disappears more rapidly from the brain. So we began to think about the cells that are actually responsible for removing the hematoma. After ICH, microglia and infiltrating macrophage are the major phagocytes responsible for the electrophagocytosis. That leads us to a new hypothesis. Perhaps CDNF does not merely suppress inflammation. Perhaps it actively changes the clearance function of microglia. So, from this point, the project moved from neuroprotection into the microglia biology. To investigate this possibility, we directly isolate microglia and myeloid cells from the hemorrhagic brain. At the selected time point after ICH, here is day 3, the hemorrhagic stratum was dissected and mechanically dissociated using the GentleMag system. We then enriched CD11b positive cells using magnetic separations. RNA was extracted from these cells for RNA sequence. This approach allowed us to ask a much more specific questions. What transcriptional changes occur within microglia after CDNF treatment? The RNA sequencing results show a substantial effect among approximately 3,000 detected genes. 1,469 genes was significantly different between the ICH serine and ICH CDNF group. So CDNF was not producing only a small change in a few inflammatory genes. It was broadly reprogramming the microglia transcriptome. This was particularly interesting because recent single-cell studies have shown that microglia populations are highly heterozygous. Therefore, rather than asking whether CDNF simply pushed cells from M1 to M2, we began to ask what functional state these microglia are actually entering. We then look at the microglia population histologically. Interestingly, by day 3, CDNF treatment reduced the number of CD11b positive cells in the perihematoma region. At first glance, one might interpret this simply as reduced inflammation. However, we analyzed the transcriptome of the remaining isolate cells. We found something very unusual. These microglia contain an unexpected high level of transcript associated with hemoglobin. So even though there were few activate cells around the lesions, those cells appear to be carrying a very different molecular signature. This slide shows that unexpected enrichment. Several hemoglobin-related genes appear strongly in the CDNF, for example, the hemoglobin BS A2, HBB, and A1. At first, this result was confusing because microglia should not normally be major producer of hemoglobin. One interpretation is contamination, but another biological, much more interesting possibility is that the isolated microglia contain RBC-derived cargo because they had engulfed more RBC. This interpretation fit very well with our earlier observation that residual hemoglobin in the brain was reduced. So we began to connect these two things. Less extracellular hematoma, but more blood-derived material within the phagocytic microglia population. We next examine markers associated with activate and disease-associated microglia. Several of these genes were altered following CDNF treatment. Importantly, the pattern did not simply resemble global immunosuppression. Instead, it was consistent with a functional reorganization of the microglia response. In other words, CDNF appeared to change what the microglia were doing, rather than merely reducing how many microglia are represented. This distinction is important for our concept of microglia reprogramming. At this stage, the key question becomes very simple. Could this microglia actually be carrying more red blood cell-derived cargo? Transcriptomics can give us clue, but it cannot by itself prove phagocytosis. So we need another direct functional assay. For that reason, we established an in vitro phagocytosis platform using primary microglia. This assay is illustrated here. We first isolate RBC and label them with the fluorescent dye, CFDA. This labeled RBC will then incubate with the primary microglia at an approximately 50:1 RBC to microglia ratio. After incubation, extracellular RBC will be removed and the amount of RBC material taken up by the microglia can be measured by fluorescent microscope or spectrophotometry. These provide a quantitative phagocytosis index. As you can see from this image, the fluorescence RBC signal can clearly be detected inside the CD11b positive microglia. So now we had a direct system to test whether CDNF itself enhanced RBC uptake. And the answer is yes. In primary microglia culture, recombinant CDNF significantly increased the uptake of fluorescence-labeled RBC. This is important because this is an isolated cell system. There are no neuron, no blood-brain barrier, and no reduction in lesion size that could indirectly expand the effect. Therefore, CDNF can directly on microglia to enhance phagocytosis. We also examined several receptors involving the recognition and uptake of damaged blood component. CDNF increased the expression of scavengers receptor, including CD36, CD163. This provides a potential molecular mechanism linking CDNF treatment to increase hematoma clearance. We then asked whether the same mechanism occurs in vivo, and the answer was again consistent. In the hemorrhagic stroke, striatum, CDNF increased the expression of several scavenger receptors, including CD163, CD36, and CD91. Neuroscience demonstrate that much of this increase occur in CD11B positive microglia and macrophage. These receptors have complementary function in recognizing and processing blood-derived material. Therefore, CDNF appear to increase not only the presence of phagocytic machinery, but also the cellular capacity for hematoma recognition and uptake. These help us explain why CDNF treatment treat the animal clear the hematoma more rapidly. But engulfing RBC create another problem. Phagocytosis is only beneficial if the cells can safely process what it has engulfed. RBC degradation release hemoglobin, heme, and iron, all of these which can generate substantially oxidative stress. Therefore, successful electrophagocytosis require not only uptake, but also an effective intracellular detoxification system. This is where the NRF2-HO1 pathway becomes important. We found that CDNF increased nuclear NRF2 activity and subsequently increased HO1 expression in a time-dependent manner. At the same time, protein carbonate level were reduced, indicating lower oxidative stress damage. So our interpretation is that CDNF provide two coordinate effects. It increased the ability of microglia to take up RBC-derived cargo, while also improving their ability to process that cargo under oxidative stress. This may be essential for maintaining effective clearance without driving excessive inflammatory injury. So this slide summarizes the main message of the study. Following hemorrhagic stroke, red blood cells accumulate within the brain and subsequently release toxic blood-derived product. With CDNF treatment, microglia and macrophage increase the expression of scavenger receptors such as CD36, CD163, and CD9. These promote electrophagocytosis and accelerate hematoma resolution. At the same time, activation of the NRF2-HO1 pathway improves antioxidant capacity and help the cells manage the oxidative burden generated during hemoglobin degradation. CDNF also reduce pro-inflammatory signaling, perihematoma edema, and secondary brain injury. Therefore, the therapeutic effects is probably not due to the one pathway alone. The other CDNF appear to coordinate hematoma clearance, inflammation resolution, and cellular stress adaptations. And this brings us back to the title of the talk, From Hematoma Burden to Brain Repair. Instead of viewing microglia as inflammatory cells that should be suppressed, our results suggest that they can be therapeutically reprogrammed into an active component of brain repair. Our broader concept is therefore not simply to suppress neuroinflammation, but to restore the capacity of microglia to clear, process, and resolve the injury. Most of the data I mentioned have been published on cell death and disease in a good journal with 12.2 impact factor. Finally, I would like to acknowledge all the people who contribute to this work. I would especially thank to the Professor Mikko Arabeara and Professor Mark Salma who discovered this CDNF. And I'm also very grateful to all of our collaborator members, students, and colleagues who contribute to the animal experiment, molecular studies, imaging, and to the data analysis. I would also like to acknowledge the funding agency that support this work. Science is always a collaboration effort, and this project would not have been possible without all of this contribution. And thank you very much for your attention. I'm very happy to take any questions. Thank you.
Thank you very much, Professor Liao, for your insightful presentation. Nice story from the usage of CDNF in activation of microglia in clearance of the hemoglobin that resolves we can see that within 3 days, right? It can resolve
Yeah.
the hematoma in rats model. So for audience, remind again, if you have any questions, you may ask Professor Liao from the Q&A box in the chat box in the Zoom. At the below right panel, you can see Q&A. You can ask Professor Liao in that box, or you maybe ask directly later on after the second speaker ends her talk. We may allow you to directly ask our speakers. So let's move. Thank you, Professor Liao. Now we move to the second speaker, which is Dr. Agriani Dini Pasiana. So Dr. Pasiana is my colleague here at the Center for Biomedical Research. And today she will be presenting her research back in Japan about prion disease. So the topic for Dr. Pasiana is breaking the chain of prion, identifying the molecular switch that drives protein misfolding. Maybe a lot of you never heard about prion disease before, because this is a quite rare also for me. So without waiting too long, let's hand the floor to Dr. Pasiana. So, Dr. Pasiana, you have around 30 minutes for your presentation. The screen is yours.
Okay, thank you very much. Can you hear my voice?
Yes, very clear.
Okay, let me share my PowerPoint first. And then, okay, can you see my PowerPoint?
Yes, you may proceed.
Okay, thank you very much. Good morning, everyone. Today, I would like to talk about a prion disease, which is a rare but very serious brain disease. And the interesting thing about prion is that the problem begins when a protein changes its form. And later, I will show you how researchers narrow this problem from the entire protein into the only few amino acids that may help control this conversion. So, what exactly is a prion? Prion is not a virus and it's not a bacteria. So, it's related to protein that is already naturally present in our brain. And the normal protein is harmless. But when it changes the form into the abnormal protein, it can encourage the other protein to change the form as well. So, this will create a chain reaction that can damage the brain. So, although the prion diseases are very rare, they are extremely serious. Once symptoms appear, the disease continues to progress, and unfortunately, there is no treatment that can stop or reverse it. So, this disease can affect humans as well as animals such as cattle, sheep, deer, and elk. And in humans, only about 1 or 2 cases per million people each year, but the outcome is very devastating. So, prion disease is actually a family of disease. In human, one of the best known example is Creutzfeldt-Jakob disease or CGD. And a cattle can develop a BSE, or also known as a mad cow disease, and a sheep and a goat can develop a scrapie, while deer and elk can develop a chronic wasting disease. So these diseases are related, but they do not always behave in exactly same way. The type of prion and the host can make an important difference. So prion research has a long history. So scrapie, a prion disease in sheep, was described 100 of years ago, and CGD was recognized in the 1920s. And Kuru later provide evidence that human neurological disease could be transmitted. And in 182, Stanley Prusiner introduced the term of prion from proteinaceous infection. And in 197, he received the Nobel Prize for that. And to this day, researchers are still trying to understand exactly how these prions change their shape. So to put the number into the perspective, CJD is estimated to affect only about 1 or 2 people per million each year. However, we do not have a complete global registry, so the true number is still uncertain. And another challenge is that the early symptom can look similar to the other neurological disease. So because of this, some cases might be missed or diagnosed as something else. So the damage, how abnormal prion disease uh, uh, happen in the brain. So the damage begin when the normal proteins convert into the abnormal protein. And this abnormal protein can collect together and form a harmful clumps. And over time, this will damage and kill the nerve cells. And eventually, the brain tissue can develop a sponge-like appearance. And unfortunately, as I said, this is progressive, and which means the damage becomes worse over time. So here I show you the pathological change in the brain specific to prion disease, including flocculation. So it's like a hole, and that's why we call it a sponge-like appearance. And there are also a neuronal cell loss and the presence of amyloid plaques. So now that we understand why prion is important, let us move from disease itself into the laboratory. So the next question is, what actually happened at the molecular level? So to answer this, we begin to looking more closely at the prion protein and the small region that may control its behavior. So there are 3 main questions behind this research. So the first, why does normal protein change into the abnormal protein? The second, are there specific parts of the protein that control this transformation? And the third, if we can identify for those control points, could they eventually help us to develop a treatment? So for your information, before we can design the drugs, first we need to understand where and how the conversion was begin. So here I show you the secondary structure of a normal PRP. So normal PRP consists of 2 major domains, the N-terminal domains, which is short from residue 23 to 120, and the C-terminal domains, which is short from residue 121 to 231. So the normal PRP is initially expressed as 254 amino acid, and the 22 N-terminal acid comprise of a signal peptide that allow normal PRP to translocate it into the endoplasmic reticulum. In the endoplasmic reticulum, the 22 N-terminal amino acid are removed by proteolysis, and at the same time, in the 22,3C terminal amino acid removed for the addition of a GPA anchor. So in the N-terminal domain, there are specific regions such as 2 polybasic region, octapeptide region, and hydrophobic region. And in C-terminal region, there are 2 beta sheets, 3 alpha helix, and 1 disulfide bond, and 2 glycosylation sites. So there are several reports indicating that N-terminal domains play an important role in the conversion of normal protein into the abnormal prion protein. So Turnbull et al. In 2012 reported that deletion of the N-terminal polybasic region reduced the sensitivity of prion infection and reduced the abnormal level in the brain, while Hara et al. In 2018 also reported that deletion of the OR region play a differential role in the pathogenesis of BSE prion and RML or 22L scrapie prions. So, the research strategy was basically to zoom in step by step. As I mentioned before, that the protein expressed 254 amino acids. So instead of studying every residue at the same time, we're looking for the small part of the residue that can induce change into the abnormal protein. So at first, we will focus on the region from amino acid 91 to 106 and then narrow it even further to only 3 amino acids at the position 97 to 99. So we hypothesized that if changing such a small region can change the behavior of the whole protein, then that region must be important. So to make the result more reliable, our team did not depend only on one type of experiment. We used animal model to observe disease in the whole living system. We also used the cell model to study the process under the control condition. And we do the biochemical methods to reproduce and amplify the protein conversion in the laboratory. And we also do the protein detection method to confirm whether the abnormal protein was produced. So together, this approach gives us more stronger picture than any single experiment alone. So now we come to the most important part. What did this experiment actually show? So later, I will show how we were able to narrow the process to a surprisingly small area of the prion protein. And this finding will help us to understand which part of the protein are important for the conversion of a normal protein into the abnormal protein. So in 2020, Uchiyama Sensei, one of associate professor in our lab, he studied what happened when the N-terminal region of the prion protein around amino acid 91 to 106 was removed. So at first, he produced a transgenic mice termed TG-PRP delta 91 to 100 PRP null mice, and then he intracerebrally inoculated four different prion strains, which is an RML, 22L, FK1, and the BSE prion into the transgenic mice, and he found that transgenic mice develop disease with a very long incubation time after inoculation with BSE prion. However, transgenic mice did not develop any symptom after inoculation with RML, 22L, and FK1 prions. So these results suggested that residue 91 to 106 might be involved in the prion pathogenesis in a strain-dependent manner. So the next study, we went one step further. Instead of only asking whether the larger 91 to 106 region was important, we want to identify exactly which amino acid inside that region were responsible. So this study was published in the Journal of Biological Chemistry in 2020. So later, we then tried to narrow down residue 91 to 104 by transfecting expression factor encoding PRP mutant with a various deletion within residue 91 to 104, such as 91 to 104, delta 91 to 96, delta 91 to 100, delta 96 to 100, delta 96 to 104, and delta 100 to 104 into the RML and 22L infected cells. So using 3F4 antibody, we found that only PRP delta 91 to 96 was converted into the abnormal protein, suggesting that residues other than residue 91 to 96 are important for the conversion. So, however, we suspect that PRP delta 100 to 104 was hyperglycosylated, suggesting that possibility that the unsuccessful conversion of PRP delta 100 to 104 is due to the glycosylation, not due to deletion of residue 100 to 104. So the Western blot analysis confirmed our suggestion that PRP delta 100 to 104 was hyperglycosylated, while PRP delta 100 to 103 was not hyperglycosylated. So we also found that PRP delta 100 to 103 was converted into the abnormal prion protein in the both cells. This is indicating that the glycosylation in the PRP delta 100 to 104 is responsible for its unsuccessful conversion into the abnormal protein, and that the residue 100 to 103 are not important for the abnormal protein conversion. So to further confirm the role of residues 97 to 99 in the conversion of normal protein into the abnormal protein, we transfected PRP mutant with a single deletion of residue 97, 98, and 99 into the RML and the 22L infected cells. So as you can see here, the result shows that PRP delta 97, delta 98, and delta 99 were not converted into the abnormal prion protein. So this result supported that residue 97 to 99 could play an important role in the conversion of normal protein into the abnormal protein in the infection with RML and 22L prions. So this is the PMCA assay. So at first, we produced baculovirus recombinant wild-type prion protein, and then the PRP delta 97 to 99, and then subjected this prion protein to PMCA with RML infected, 22L infected, FK1 infected, and the BSA infected mouse brine homogenates. And as a control, we use prion uninfected brine homogenates. The result shows that PK-resistant prion protein was generated from wild-type prion protein with RML-infected mouse brine homogenates. However, PK-resistant signal were not observed from PRP delta 97 to 99 with RML-infected mouse brine homogenates. The same results also obtained with 22L infected and FK1 infected mouse brine homogenate. In a contrast, wild-type prion protein and PRP delta 97 to 99 were converted into the PK-resistant prion protein with a BSE-infected mouse brine homogenate. So these results suggested that residue 97 to 99 are important for the conversion of normal protein into the abnormal protein in the infection with RML, 22L, FK1 prions, but not with BSE prions. So as summary here, we found that residue 91 to 106 are crucial for normal prion protein to convert into the abnormal prion protein after infection with RML, 22L, and FK1, but not with BSE prion in mice. And we also found that residue 97 to 99 are particularly important for normal prion protein to convert into the abnormal prion protein in an infection with RML and 22L prions. And in vitro PMCA assay also support the important role of residue 97 to 99 in the conversion of normal protein into the abnormal protein in infection with RML, 22L, and FK1 prions, but not with BSE prions. So you probably wonder, like, could this discovery lead to the treatment? Well, possibly, but we need to be careful. Finding a molecular control point is one first step. So the result must be confirmed, the mechanism must be understood, and the researcher then need to find a safe way to target it. After that, there will be a preclinical studies and eventually clinical trials in patients. So I can say that this research is still at the discovery stage, not yet at the treatment stage. So before I finish, I will like to leave you with a few key points. First, prion disease involves proteins that change into the abnormal forms and can induce other proteins to do the same. Second, this disease mainly damage the brain and the nervous system. Third, they are very rare, but they are very serious, and unfortunately, until now, there are treatment for that. And prion disease can arise in many different ways. They may be inherited, acquired, or occur spontaneously. And finally, this research is extremely important because understanding how prion behave is the first step toward better diagnosis, prevention, and eventually a treatment. So our study contribute to this effort by identifying a very small part of the protein that appears to play an important role in the convergence process. So this research will not have been possible without the help of multiple labs, numerous collaboration, and a long-term discussion with a professor and friends. So I would like to say thank you very much. And that brings me to the end of my presentation. So I hope this talk will give you new information, knowledge, and show you how studying something as small as 3 amino acid can help us to understand a much larger and a very serious disease process. So thank you very much for your attention.
Thank you very much, Dr. Pasiana, for your interesting presentation and insightful story about the prion disease and your research back in Japan about prion disease, where you find that the region from 97, right, to
Yes.
99 is the molecular switch for this prion disease. So it's very interesting that prion disease is actually an infectious disease, right? But it is not a live agent, right? It's a protein.
Yes.
Yeah. It's not bacteria, it's not virus. It's a protein, but it is infectious. Very interesting.
Yes.
So we have several questions here addressed to both of our speakers. We have questions from our college, Dr. Novaria Panjaitan, also from Dr. Putu Yulandari, also our college, and from Dr. Feni Betriana, our college, and also from YouTube. We have also several questions from YouTube. Let me ask first Professor Liao about a question from Dr. Novaria Panjaitan. Yeah, very interesting questions, and also similar question from other researchers. So the questions is, it is interesting that CDNF is increased after ICH, right? But
Yes.
it is very interesting at the early stage of ICH, the level of CDNF is lower, right? Is significantly lower. Can you give us insights, Professor Lea, on how this might happen?
Yes. One possible the explanation is the severe disruption of protein homeostasis. During the acute phase of the injury, everything is activated, including some protease and the oxidative stress that will damage the protein and causing the protein degradation increase. So that's why we see that during the acute phase, the protein level of the CDNF is decreased. Yep.
Oh, I see. So this CDNF, what neurons or what cells that produce this, secrete this CDNF then?
Mostly it's dopaminergic neuron from the beginning that the Professor Max Arma discovered the CDNF from the dopaminergic neuron. But nowadays we don't know what is the CDNF receptor until now, even though already got 20 to 30 years ago. We still cannot find the receptor of the CDNF. We just know that the CDNF can penetrate, go inside, produced by the cells, and then act some ER retention proteins.
Okay. So from your finding that CDNF, can I say that it is specifically targeted microglia when it is activated, or?
No, they also can act to neuron and oligodendrocyte and astrocyte also. But we just focus on the microglia. Yeah.
I see. Okay. Thank you for your answer, Professor Liao. Now let's move to the question for Dr. Pasiana. So we have question here, similar question from several people. Let me wrap it for you. So we have similar question from Dr. Uli and our anonymous attendee here. The questions is why this BSE behave differently from RML22L or FK1, and why did you use this? And yeah, I remember BSE, although the mice, right, you use transgenic mice, although it is resistant to the other, but to this BSE, it is in fact, it is show some prone, right? Although is the response is lower. Can you give us insights, Dr. Pasiana?
So why you asking like why does BSE behave differently and
Yes.
why I use another size? So
Yeah.
actually, for me, is also this is the most interesting findings of our study. So when it's showing that PRP deletion residue 97 to 91 could not be converted into the RML and 22L or FK1, while BSE could convert it into the abnormal PRP. So this is indicated that molecular requirement for this conversion is strain dependent manner, but it's not a universal switch for all the different strains. So I studied these different strains to see if all these strains have a same molecular mechanism.
Okay. So these different prions, can I say they are prions?
Yes,
These-
different strain.
Yeah, different strains. So these different strains of prion is isolated from different species or the same species like BSE, RML, 22L, FK1?
Yes, from different species. BSE is a mad cow disease.
Oh, Medco disease. Okay.
Yes.
So can they all infect human?
No, different strains. So for BSE is for mad cow disease. It's for cow, and the scrappy is for the lamb and a goat. And for humans, it's known as a CGD, Crawford-Jakob disease. So I don't do the strain for the human yet, only for the animals.
Okay, interesting. I have similar question here we have similar question here from Dr. Reni Patmasari from Universitas Muhammadiyah Makassar, addressed to you also, Dr. Pasiana. She asked, can this prion disease spread from one person to another? And I also have similar questions. How do you get this prion disease? Does it spontaneously happen in your body or any other demographic people that prone to disease, for example, older people or younger people or what? Can you give us that? Thanks. Seems your voice is muted. Yeah. Okay.
So the question is, can this disease spread from one person to another person? So Yes, as I mentioned before, this disease can be inherited and
Mm.
spread to another person is not. So this is a protein, so it's not like a virus. It's not like a COVID-19 that it's spread through the coughing to the air, but it needs something to transmittable, such as foods or medical care centers. And so yes, it can be spread, but it needs something to move it to another person.
Okay,
I hope
so
you
it need
understand
to
what it
move
means.
it need to move into your brain, isn't it? Doesn't it?
Uh, Mostly prion is found in the brains, and it's already in your brain. So the infectious protein it will be come into your brain and induce another normal protein to also misfold it, the prion protein inside your body.
Okay, I see. Thank you, Dr. Pasiana. And now we move back again to Professor Liao. We have an
Yes.
interesting question here from Sigit ICT. Yeah. He asks about one of your paper. Yeah. He
Yes.
asked about your work on immunoproteasomes and microglial clearance. And do you think combination of these can enhance could be a promising strategy to improve the hematoma?
Yes, I think that is very interesting and mechanistically reasonable strategy. But the key issue would be the timing and the balance. Based on our work, I think the immunoproteasome inhibition may help reduce excessive inflammatory signaling and shift microglia away from a strongly pro-inflammatory state. However, the hematoma resolution more uh, require more than reducing inflammation. Microglia also need sufficient phagocytic, lysosomal, and metabolic capacity to internalize and degrade erythrocytes, hemoglobin, and other blood-derived cargo. So in principle, combining immunoproteasome modulation with enhancement of lysosomal function could provide two complementary effects. One arm reduce maladaptive inflammatory signaling, while the other restore the intracellular clearance machinery. I think I would see the optimal strategy not simply as proteasome inhibition plus lysosome activation, but as a broader proteostasis and immunometabolic reprogramming approach, reducing excessive inflammatory signaling, preserve mitochondrial fitness, or improve lysosomal degradation, and then restore microglial phagocytic capacity and accelerate hematoma resolution. Yeah.
Then the following up, do you think that adding CDNF into this can be successful? I mean,
You mean
combining CDNF with immunoproteasome or enhancements of lysosomal function.
there's a little bit complication because the immunoproteasome inhibitions is some similar like they overactivate, especially the form of the proteasome. Then if you plus another drug, for example, the CDNF is quite a little bit mixed up. We don't know which affect which one. So I think this is maybe can try later on, but I think that the timing also is the very important issues that we need to consider.
Yeah, because from your presentation that using omics, right? Using omics, you notice that this microglia is diverse, right? They are not like kind of M1 or M2. While using immunoproteasomes, they can direct this microglia towards M1 phenotype. So I think, okay, maybe we combine the CDNF, maybe enhance. That's just my thinking.
Yeah.
Okay. Thank you, Professor Liao. We have another question here from our colleagues also. Let me see again the questions. Yeah. You answered that. But
Yeah.
let me reiterate this question again. So from Dr. Wiwit Nurwidianingtyas, a researcher also in our center, she asked that given that these microglia act as the brain first responders, right, and play an essential role in this initial inflammation and protective response following ICH, and why should they consider a therapeutic target? And more importantly, what's the optimal time window, as you mentioned before, for this targeted therapy? 1 hour, 2 hour, 3 hour, and which clinical manifestations, image finding, or biomarkers could help identify the appropriate time to intervene?
I think the suitable timing for interventions to administrate the CDF is, I think, is much more early as early as possible. That's why we designed the experiment during the acute phase. Then we just administrated the CDNF inside the brain, because this concept is based on we need to reprogramming the microglia. During the acute phase of injury, the microglia will become much more overactivated and towards pro-inflammatory level stage. So we need to switch to prevent or to slow down, to reduce the inflammation and promote much more phagocytic and anti-inflammatory property of the microglia. Switch the microglia to become much more anti-inflammation status to calm down everything, and then hopefully can clean and clear the debris and also the rest oxidative stress and the debris. Yeah.
Okay, since this CD and F, right? When you apply this in ICH, in hematoma, we can see that these microglia, as you show in omic data, they are express some of the hemoglobin genes, right? Hemoglobin genes. Have you maybe thinking or already research about the effect of this CDNF on another cells such as astrocytes or even neurons? Will they show similar patterns to this CDNF treatment?
Okay, first of all, we isolate the microglia and we found the hemoglobin genes within the microglia. Just like I emphasized that this is not the microglia produce these hemoglobin genes, but they engulfed the RBC. That's why the microglia, what the microglia do, they will engulf, find, eat, and digest. So this is the process. The microglia already engulf the RBC, try to engulf and digest this RBC. And another question is the CDF experiment already have been long focused on the dopaminergic neuron, which was carried on by the Professor Mark Sama in Finland and Sweden. They already pushed this drug as to, I think, clinical trial phase 2 already. They almost reached the phase 3 stage. They focus on to prevent the dopaminergic neuron dying from the Parkinson's disease. Yeah, I think they also act on the neuron also.
Okay. But not necessarily shows similar hemoglobin expression, right? On another.
No. They're totally different because neuron didn't produce any hemoglobin. Actually, the cells, neuronal cells, doesn't produce any hemoglobin like the genes within the cells. Only microglia after ICH, they engulf and try to clean the debris. Then they produce after isolate. Then we find the hemoglobin genes within the cells.
Not even astrocyte. Not even astrocyte.
No, we didn't check for the astrocytes.
Okay.
We just focus on microglia.
Okay. Thank you, Professor Liao.
Yeah.
We have another question here from Dr. Mardiana, Meti Mardiana. She asks you, the CDNF takes 3 days, right? In your rat model. So she thinks that this is less suitable as a replacement for alteplase, which is the standard pharmacotherapy for the ICH during the golden hour of stroke treatment. Yeah. How does it compare to current use of antithrombotic such as clopidogrel or citicoline as a neuroprotector?
Actually, it's a total different story because they asked the way is the ischemic stroke. Ischemic stroke got the golden period for about 3 hours to 4.5 hours. Try to lysis the clot. But in hemorrhagic stroke, there are currently no any treatment because the hemorrhage is bleeding within the brain. Only surgical treatment within the severe patients. Otherwise, in moderate and mild disease of the patients, they only for handle for the supportive care. That means that just calm down and not this is limitation of the, but no matter it's severe or the moderate, the blood clot remain. That's why we focus on the way that clean and clear the debris, clot debris.
So you say that even after this golden hour of ICH, this CDNF treatment might be possible also, right?
Yes, actually in ICH, there's no such golden period.
Okay.
Yeah.
Okay, so there is no golden, but since after the ICH, they still show some symptoms, right? Due to the hematoma in the brain. Okay, I see.
Yeah.
Thank you, Professor Liao. Let's
Yeah,
move
okay.
back to Dr. Pasiana. We have many questions addressed to you. Very interesting. Yeah. About prion disease. Okay. Let me check again. Yeah, from your presentations, I also feel that some technical maybe need more explanations. We have a question here from anonymous attendee. Can you explain again about the cell-based assay and the PMCA? Why you use this? I mean, maybe they think that why not directly in animal, right? Why use cell-based? And can you give us insight why?
So, using cell-based experiments will allow us to examine the PRP conversion in a biological cellular environment. And however, cell system usually contain many cellular components, so that could potentially influence the results. So while PMCA provides an in vitro biochemical approach to examine the conversion process more directly. So I using these two analysis because these two systems will provide a complementary information.
Okay, so you also use mouse, right? You mentioned that you didn't in your research back in Japan you didn't use human, right?
Yes.
And you just use animal. And following up from Dr. Feni Betriana, why did you use mouse PRP, right? If you research in mouse, since they are different in human, can this be applied? Your research in mouse and cells, can this be applied in human also?
Probably. Well, I'm not really sure yet because as we can see here, the results also in the as I conclude before that even in the mouse it was a strain dependent manner. So different strains will be show a different results, such as using 97 to 99 BSE not converted into the abnormal but 22L, RML, and FK1 was converted. So I'm not really sure if we're using human cells, it will convert it or not converted into the abnormal prion protein.
Okay, that's why this also following up your answer. There is a question also from anonymous entity. That is why the BSE results, it seems contradict the idea that residues 97 to 90 are molecular switch.
So it's as I mentioned, it's not. I cannot say it's contradict, but we can say that the result is strain dependent manner. So different strains will be give a result we will be given a different results. BSE is converted, but FK1 22L and RML is not converted.
Okay, so this different strain, right? The prion strain, I can say the prion strain are the arrange of the amino acids conserved.
Can you repeat?
Are they conserved the arrange of amino acids between prions? Are they similar?
I'm sorry, I don't understand. Can you?
Ya, maybe I switch to bahasa Indonesia. Ya, jadi ada beberapa prion kan ya, Dr. Anggi. Ada beberapa prion strain. Jadi susunan asam amino dari beberapa prion ini apakah similar gitu, apakah sama? Ya.
I think it should be same. It should be same between all the strains. But why it's have a different results between the BSE and and another strains? It's still unknown why it's have a different.
Okay,
But
so
if we
actually,
talk,
yeah, this prion actually, even though they are from mice, from cow, from cattle, from deer, actually they are the same thing. They are the
yes,
same
it's the
prion.
same prion. Yes.
So what makes they are different? What make they different between all
What's
those? Yeah, what make these prions different? Even not even a single amino acid different between those prion?
I'm not really sure for that question yet, since I didn't search for them. Are we talking about the molecular structure here?
Yes, that's right. That's right.
Yes. So for that, I'm really sorry. I didn't do that kind of experiment yet. And as far as I know, also there is no research yet to confirm whether all the type of prion strain have a different molecular structure. So I'm really sorry I can answer that question.
Okay. Thank you, Dr. Anggi. Yeah. Thank you for your answer. Yeah. We still have questions here. I have questions here for Professor Liao. It is interesting that you mentioned that during ICH there is a lot of iron release, right, in the brain. So I think that since a lot of release of iron, I think maybe it shows some similar features to ferroptosis. And do you think that ferroptosis, some pharmacotherapy can be applied to this ICH?
Actually, ferroptosis is one of the death pathway after ICH, because ICH will trigger a lot of the different ways of the injury. For example, the ER stress, the autophagy, and necrosis and apoptosis also. I think, yeah, ferroptosis target on ferroptosis, for example, using some chelator. For example, the ferrous chelator is one of the, also is a very good options, good strategy to reduce the injury after ICH. But I think we didn't study the CNF affected the ferroptosis pathway. We already know that they focus on the ER stress and also the apoptosis.
Okay. Okay. Have you considered to do your research using the anti-ferroptosis agents in the ICH? Since I think maybe the ferroptosis is the upstream of this pathway.
Yeah, I totally agree with you, because we got some students focus on using the chelator and target on the ferroptosis injury after the injury. Yeah, we straightforwardly inject the chelator within the brain using the severe IHH model.
Okay, interesting. Maybe we invite you again to talk about this again.
Yeah, yeah, but pressure. Yeah.
Another question. Yeah. I have a question for this CDNF. In the RETS model, did you give the CDNF only once or repeatedly?
We only intracerebrally inject one. We also got another experiment through the systemic administration by intranasal and also the intraperitoneal also. Yeah, they all show the similar beneficial effect in reducing the ICH injury.
Okay, so the CDNF can penetrate the BBB. I see.
Yes.
Yeah. Let me check again if there is any missed question, or is there any audience who wants to ask directly to our speakers? You may raise your hand directly. We welcome you to directly ask our speakers. Is there any? Yeah, seems there is no one who wants to ask directly. Yeah. Okay. Let me check again if there is any questions to any of you. Yeah, seems there is no questions again, or any question from our panelists? No? Okay, since there is no more questions, we can wrap our session today, and a very thank you to both of our speakers for their excellent talks, give us insight of how microglial clearance can resolve the hematoma and how prion disease is infectious disease, and also hopefully we can cure the prion disease. So thank you for both our speakers. The next is I may hand the floor over to Bu Rindrin. Are you here?
Yes.
Yeah. Thank you.
Oke, terima kasih. Ya. Baik, terima kasih Jiro Hasigawa Sitomora yang telah memandu sesi pemaparan dan diskusi yang sangat menarik ini. Dan kepada seluruh narasumber, kami mengucapkan terima kasih atas materi yang telah disampaikan. Semoga menjadi pencerahan bagi kita semua. Baik, Bapak Ibu, bentuk apresiasi kepada narasumber dan moderator, kami akan memberikan sertifikat yang akan ditampilkan di layar oleh panitia. Baik, kami mengucapkan terima kasih kepada narasumber dan penyerahan sertifikat pertama kepada Profesor Leok Hokkien, Research Profesor dari Department of Medical Research, Hualien Tsuji Hospital. Sertifikat akan diserahkan secara virtual oleh Kepala Pusat Riset Biomedis Profesor Sunarno atau yang mewakili Badan Riset and Tropical Research kepada Ibu Ana Listia, disilakan.
Hello, Professor Liao. On behalf of the head of our research center, who unfortunately could not join us today due to an unexpected meeting, I would like to present this certificate to you virtually. And thank you so much for being our speaker and for sharing your valuable knowledge and insight with us. We truly appreciate your time and contribution. And so thank you once again, and we hope to see you again, or maybe even a collaboration together with you in the future.
Yes, it's a pleasure. Thank you.
Oke, thank you. Ana Listia. Penyerahan sertifikat kedua ke Pasiana PhD. Peneliti ahli muda Pusat Riset.
Terima kasih Mbak Rin. Dr. Agriani Pasiana, terima kasih mewakili Pak Kapus yang berhalangan hadir juga. Terima kasih atas waktunya, dan Berikut adalah sertifikat sebagai bentuk apresiasi atas kesediaan Dr. Agriani Pasyana sebagai narasumber pada webinar hari ini. Semoga ilmunya dapat diserap oleh para hadirin dan peserta webinar hari ini. Terima kasih, Dok.
Terima kasih Mbak Ana.
Terima kasih Mbak Anda. Selanjutnya penyerahan sertifikat kami berikan kepada moderator hari ini, Jiro Asigawang Sitomorang PhD, peneliti Pusat Riset Biomedis OR Kesehatan BRIN. Kami
Ya, Dr. Jiro, terima kasih. Mewakili Pak Kapus juga.
kembali kepada
Ya.
Ibu Ana untuk memberikan sertifikat.
Oh, mohon maaf, Mbak Rini. Terima kasih atas waktunya Dr. Jiro telah menjadi moderator hari ini.
Ya, terima kasih, Mbak Ana.
Ya.
Baik, terima kasih Ibu Ana Listia mewakili Bapak Kapus hari ini. Baik Bapak Ibu serta kawan BRIN yang berbahagia, alhamdulillah kita telah sampai di penghujung acara. Terima kasih kepada seluruh peserta yang telah menyimak acara ini dari awal hingga akhir. Kami mengingatkan kembali kepada Bapak Ibu untuk mengisi presensi tautan yang telah di-share panitia. Bisa dicek atau diunduh pada tautan www.sbrin.co.id sertifikat 1 September 2026. Saya Rindri Lastriani, mewakili petugas yang bertugas hari ini, menyampaikan mohon maaf jika ada kekurangan dan kesalahan dalam acara berlangsung. Izinkan saya menutup dengan sebuah pantun. Bunga melati harum sekali, tumbuh rapih di tepi taman. Terima kasih peserta yang sudah hadir di sini. Semoga ilmu menjadi bekal di masa depan. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih dan salam sehat semuanya.
Terima kasih.
Terima kasih. Thank you very much, Profesor Liao.
Thank you very much. Yeah, you may leave, Dr. Ranggi. Thank you very much.
Thank you.
Riset
Thank you,
adalah kekuatan untuk kita bisa menembus segala keterbatasan. Inovasi adalah visi yang kita segerakan untuk masa depan. Kita tidak bisa sendiri. Kita harus bersama untuk membuatnya menjadi nyata. Kita tumbuh di bumi Indonesia, tempat yang magis dan penuh keajaiban. Terbentang di antara dua benua dan samudra, kaya akan keanekaragaman kultur dan biodiversitas. Indonesia dan kita adalah paduan yang dibutuhkan untuk mewujudkan visi besar di masa mendatang. Layaknya sebuah orkestrasi, Badan Riset dan Inovasi Nasional atau BRIN hadir mengharmonisasikan berbagai kegiatan penelitian, pengembangan, pengkajian, dan penerapan, serta mensinergikan riset dan inovasi di Indonesia. Membentuk ekosistem yang terdiri dari 7 kedeputian, 12 organisasi riset, dan 85 pusat riset yang diantaranya Organisasi Riset Penerbangan dan Antariksa, Organisasi Riset Tenaga Nuklir, Organisasi Riset Energi dan Manufaktur, Organisasi Riset Kebumian dan Maritim, Organisasi Riset Hayati dan Lingkungan, Organisasi Riset Elektronika dan Informatika, Organisasi Riset Ilmu Pengetahuan Sosial dan Humaniora, Organisasi Riset Arkeologi, Bahasa dan Sastra, Organisasi Riset Kesehatan, Organisasi Riset Nanoteknologi dan Material, Organisasi Riset Pertanian dan Pangan, Organisasi Riset Tata Kelola Pemerintahan, Ekonomi, dan Kesejahteraan Masyarakat. BRIN juga mendorong riset dan inovasi menjadi ekosistem di Indonesia yang maju dan berkembang dengan 2 sasaran utama. BRIN menyadari potensi luar biasa putra-putri bangsa dalam bidang ilmu pengetahuan dan teknologi. Untuk itu, berbagai kebijakan riset dan inovasi diarahkan untuk menghadirkan kompetensi SDM iptek terbaik. Menghasilkan berbagai program unggulan sebagai bentuk komitmen BRIN terhadap periset, inovator, serta masyarakat luas dari berbagai disiplin ilmu di seluruh negeri. Peningkatan kualitas ekosistem riset dan inovasi di Indonesia juga tercipta melalui kolaborasi dengan berbagai kementerian, lembaga, industri di dalam negeri, dan pemerintah daerah. Di antaranya, dengan dibentuknya Badan Riset dan Inovasi Daerah atau BRIDA. Selain itu, kerjasama dengan berbagai lembaga riset di luar negeri juga terus didorong melalui partisipasi aktif sebagai anggota dalam berbagai organisasi dan forum internasional. BRIN menata alur riset sehingga lebih efektif dan efisien dengan mengintegrasikan sumber daya manusia, infrastruktur, dan anggaran dari berbagai lembaga litbang jirap. Berorientasikan prioritas riset nasional, BRIN melakukan evaluasi secara komprehensif untuk setiap percepatan yang tengah berjalan. Upaya tersebut didukung melalui pengelolaan berbagai fasilitas riset dan inovasi yang tersebar di 17 provinsi di Indonesia dan akan terus bertambah mengikuti perkembangan kemajuan iptek.